Continuation of the previous video, we were talking about therapeutic agents of HIV. At present, there is no complete cure for AIDS. Primary treatment is directed at reducing the viral load and disease symptoms and at treating opportunistic infections and malignancies. The antivirus currently approved to use in HIV disease are of two types. Reverse transcriptase inhibitors are nucleoside analogs that inhibit the disease reverse transcriptase as it synthesizes DNA from RNA. Examples include AZT or pseudovidine, ritrover, didanosine, Videx, then salcitabine, HIV-ID, then studine, then lamivudine, then delaverdine, and nevirapine or virumone. I'm not sure about the pronunciation. I prefer just read this and memorize the what is called as the spellings so that you can reproduce this stuff during exams. Then protease inhibitors work by blocking the activity of HIV protease and this interfere with virion assembly. Examples include idinivir, and other things. So protease inhibitors are the reverse transcriptase inhibitors. However, the most successful treatment approach is to use drug combinations. An effective combination is a cocktail of AZT, lamivudine, and a protease inhibitor such as a ritonavir. Hepatitis is a liver inflammation commonly caused by an infectious agent. Hepatitis sometimes results in acute illness followed by destruction of functional liver anatomy and cells, a condition known as cirrhosis. Hepatitis due to an infection can cause chronic to acute disease and some forms may lead to liver cancer. Although many viruses and a few bacteria can cause hepatitis, a restricted group of viruses is often associated with liver disease. Hepatitis viruses are diverse and none of these viruses are genetically related, but all infect cells in the liver causing hepatitis. Characteristics of viral hepatitis. Hepatitis A, B, C, D, E type is there. The genome of hepatitis B virus, virus, is among the smallest known of all viruses. Like retrovirus, hepatitis B virus uses reverse transcriptase using replication cycle. However, unlike retroviruses, the DNA genome of hepatitis B virus is replicated through an RNA intermediate, the opposite of what occurs in retrovirus. And plant viruses. So, the genome of hepatitis B virus is among the smallest known of all viruses. However, unlike retroviruses, the DNA genome of hepatitis B virus is replicated through an RNA intermediate, the opposite of what occurs in retroviruses. So plant viruses are also there. Tobacco mosaic virus. And prions and viroids. Prions are proteinaceous infectious agents that are responsible for neurodegenerative diseases. So, I skipped the plant viruses part. Plant viruses exist in rod and polyhedral shape. Most plant viruses have genomes consisting of a single RNA strand of the plus type. The best known plant virus is the rod-shaped tobacco mosaic virus or TMV. Relatively few plant viruses have DNA genomes. There are only two classes of DNA plant viruses. The cauliflower mosaic virus belongs to first class that contains a double-stranded DNA genome in a polyhedral capsule. The second class of DNA plant viruses contain the Gemini virus, Gemini twins. Oh, that's a new thing. Characterized by a connected pair of capsids, each containing a circular single-stranded DNA molecule of about 2,500 nucleoids. In tobacco mosaic virus, TMV causes leaf molting and discoloration in tobacco and many other plants. It was the first virus to be discovered by Dmitry Vanovsky, that first virus to be isolated, W. Stanley. TMV is a rod-shaped particle. Capsome is arranged in a hollow-right-handed helix. TMV single RNA strand is coaxially found within turns of coat protein helix such that 3 nucleotides are bound to each protein subunit. And next, prions are viroids. Prions are proteinaceous infectious agents that are responsible for neurodegenerative disease in animals, including human. Prions are unprecedented infectious pathogens that are devoid of nucleic acid and seem to be composed exclusively of a modified isoform of PRP designated PRPSC. The normal cellular PRP, denoted PRPC, is converted to PRPC through a process whereby a portion of its alpha helical and coil structure is refolded into a beta sheet. The structural transition is accompanied by profound changes in the physicochemical properties of PRP. Prions are novel transmissible pathogens causing a group of invariably fatal neurodegenerative disease that can be perpetrated by inoculating animal with the tissue extract from infected one. Prion diseases of humans are transferred as Creutzfeldt-Jakob disease, fatal familial insomnia, fatal sporadic insomnia, and Curu. In animals, the prion disease are called scrapie of sheep and goats, bovine spongiform encephalopathy (BSE), and chronic wasting disease of mule deer. Curu is the most naturally occurring subacute spongiform encephalopathy of humans, shown to be caused by prions. It was first described by the scientist Zigas in 1957. This disease is characterized by cerebral ataxia and a shivering like tremor that produces complete motor incoordination. Viroid and virusoid. An infectious agent of plants that is a single-stranded covalently closed circular RNA not associated with any protein. The RNA does not code for any proteins. Viroids have so far been shown to infect plants only. Virusoid are satellite nucleic acid. Satellite nucleic acids are single-stranded DNA or RNA, and double-stranded RNA satellites associated to viruses of the same type of nucleic acid. Most of the characterized satellites are associated with plant viruses. At most are single-stranded RNA. Satellite RNAs are always functionally dependent on specific helper viruses and are encapsulated by the coat protein of these helper viruses. Virusoid contain viroid-like circular RNA that possess ribozyme domain. Viroids and virusoids both replicate via rolling circles. The strand of RNA that packaged into the virus is called the plus strand. Both form a hammerhead structure that has a self-cleaving activity. So that's all about this microbiology portion. Later we will be having summary sessions and in topic discussions of some important topics, and I hope it will be possible for me to cover all these topics along with a quick summary of all this microbiology portion. Thank you for listening.